Translocation involving the phloem is the transport of assimilates up and down the plant to storage organs and growing parts of the plant. How are assimilates loaded into the phloem? So at the source, for example the leaf, the H+ ions in the companion cells are actively pumped out using ATP produced in cellular respiration of many mitochondria in companion cells. This creates a H+ ion gradient, so H+ ions move back into the companion cell by facilitated diffusion using a co-transporter protein. Assimilates such as sucrose move in with the H+ ions against their concentration gradient, and then assimilates diffuse into the sieve tube via the plasmodesmata. Continuing from this, assimilates lower the water potential in the sieve tube, so water enters by osmosis, causing the pressure to rise, which causes mass movement towards the sink, where the sugars are used for storage or growth when they are removed from the sieve tube by surrounding cells by diffusion. Movement of sugars in the phloem. They go from the source to the sink by mass movement, passing through the sieve plates down the pressure gradient because the hydrostatic pressure at the sink is low and there's unloading at the sink. The role of a sink is that it unloads assimilates, so it increases the water potential, causing water to move out of the sieve tube by osmosis, which creates a pressure difference. It creates a pressure gradient, so there's less pressure in the root than the leaf, causing the liquid inside the sieve tube to flow down the pressure gradient by mass transport from the leaf to the root, so the source to the sink. So sucrose is used for respiration or metabolism. Strengths for the mass flow hypothesis are that there's evidence that the phloem is used because you can use radioactive isotopes to mark glucose and use autoradiography to trace the resultant sucrose through the phloem. And if steam is used to kill a ring of bark on the tree, it stops movement in the phloem and not the xylem, and sucrose can't pass through the cut, so it builds up, lowering the water potential, so water moves in to the tissues by osmosis, causing them to swell. When aphids feed off phloem tubes, they exude sap at the other end of their body, so this is evidence that there's high hydrostatic pressure in the phloem, and aphids are usually beheaded, but the stylet still exudes sap which can be analysed, and it's shown that it's rich in sucrose and there's traces of ATP. There's also evidence that it needs ATP because metabolic poisons such as cyanide stop the enzyme ATPase working, which stops translocation, therefore it shows that it's an active process. However, the weaknesses are that there's uncertainty in the exact role of sieve pores. It was considered essential for them to have a role. The measured hydrostatic pressure is not as high as calculated to drive mass flow, and it doesn't explain why there's bidirectional movement of assimilates in the sieve tube, and it doesn't explain the movement of assimilates at different speeds in the sieve tube. And that's translocation and the mass flow hypothesis. Thanks for watching.